Hello everyone, I am Payal Kantade, MSc first year student of microbiology. I am here to give you seminar on the topic of centrifugation and its techniques. Now first here we see about the centrifuge machine. A centrifuge is found in every biochemistry laboratory. It is used primarily for the preparation of biological samples. It is used for the analysis of the physical properties of the biomolecules, organelles, and cells. The most centrifuge techniques fit into one of two categories. The first analytical ultracentrifuge developed by Theodor Swedberg in 1926. He described a new method of ultracentrifugation of producing colloidal particles and gave convincing evidence of the validity of the theory on the Brownian moment. Centrifuges were first used in the mid 14th for the process such as separation of milk. These early machines were hand driven and could reach speeds up to 3000 rpm. In 1912, new centrifuges were developed which ran on electrical power. What is centrifuge and centrifugation? A centrifuge, it is a device used for separating insoluble particles from a suspension according to their size, shape, density, viscosity of the medium, and rotor speed. The two particles of different masses will settle in a tube at a different rate in response to centrifugal force. Centrifugation. Uh, in centrifugation, any object or particle moving in a circle at a steady angular velocity is subject to an outward direction force. Directed force. The magnitude of this force depends on angular velocity in radians and the radius of rotation in centimeters. In this centrifugation process, uh, it is used to separate two immiscible liquids. The more dense component of the mixture migrates away from the axis of the centrifuge, while less dense components of the mixture migrates toward the axis. Sedimentation. It is the motion of molecules in solution or particles in a suspension in response to an external force such as gravity, centrifugal force, or electric force. Centrifugation increase the rate of sedimentation by spinning the samples and creating a centrifugal force acts on particle in an instrument called as a centrifuge. Here are some components of centrifuge. First one is motor, which give rotator motion to the centrifuge machine. Drive shaft, it is attached to motor. Rotor placed on drive shaft having space to place tubes. Centrifuge rotor. A rotor is the rotating unit of centrifuge which has fixed holes drilled up at an angle. Test tubes are placed inside these holes and the rotor spins to aid in the separation of materials. Here are some parts of centrifuge machine, that is lead, rotor, sample, motor, control unit, chamber, and latch. Basic principle of centrifugation. A particle, whether it is floating, solid, or a precipitate, or a molecule, macromolecules, or a cell organelles, is subjected to the centrifugal force when it is rotated at a high rate of speed. Centrifugal force is depend on three variables, that is mass, speed, radius. The speed is expressed in revolution per minute, that is rpm. The generated centrifugal force can be measured in terms of Earth's gravitational force as g, and the centrifugal force is denoted as RCF. Relative centrifugal force is the measurement of the force applied to a sample within a centrifuge. Centrifugal force is measured as G or xg gravity. An RCF of 500 xg indicate that the Centrifugal force applied is 500 times greater than Earth's gravitational force. R is the radius in centimeter, measured from the center of the centrifuge axis to the bottom of the test tube shield. This can be calculated from the speed rpm and the rotational radius in centimeter using the following calculation. RCF is equal to 1.119 into 10 to the power minus 5 into rpm square into r. There are three types of centrifuge machines or centrifugation on the basis of their operating system. The first one is desktop centrifugation, second one is high speed centrifugation, and third one is ultra centrifugation. But here we see about third type of centrifugation, that is ultra centrifugation. There are two main types: preparative centrifugation and analytical centrifugation. Preparative centrifugation, it is the preliminary step to separate component for further separation and analysis. It can handle large amount of samples. It is divided into two types, that is density gradient centrifugation and differential velocity centrifugation. And second one is analytical centrifugation. We can analyze the sample to small amount is used few microgram per liter to 1 ml. Mostly ultra centrifuge is used. Now let's talk about ultra centrifugation. It is used for separation of very small particle, that is virus, proteins, nanoparticles, etc. due to their very high speed. Detailed biochemical analysis of subcellular structure and biomolecules. Preparative ultra centrifuge has 6 lakh gram centrifugal force. Analytical ultra centrifuge, it is optical system to monitor movement of a particle in a centrifugal field to minimize the generation of high temperature. Rotor chamber is sealed and evacuated and refrigerated. Heavy armor planting for protection of user. It has auto temperature regulation, chamber locking, overspeed control, imbalancing prevention, etc. The first ultracentrifugation coined by Theodor Swedberg. It is the most sophisticated instrument. It operates at high speed, at speed of 75,000 rpm, providing the centrifugal force of 5 lakh gram. The ultracentrifuge is a centrifuge optimized for spinning a rotor at very high speeds, capable of generating acceleration. Intense heat is generated due to high speed, thus the spinning chamber must be refrigerated and kept at high vacuum. First type of ultracentrifugation is preparative ultracentrifugation. It is used for separation of subcellular structure and biomolecules, or it is generally used for biological materials for subsequent of biochemical investigation, that is cell organelles, proteins, nucleic acid, polysaccharides, liquid droplet or other living particles. It handle large liquid volume without any optical readout system. In 1940s, technical refinement of a preparative centrifugation technique by Cloud and colleagues positioned centrifugation technology at the center of biological and biomedical research for many decades. Preparative centrifuge designed by Pickett in 1949. 45,000 to 70,000, it is the speed of our relatives. It is the speed of preparative ultracentrifugation. Their centrifugal force is 9 lakh gram, up to 9 lakh gram. It has a their chamber is sealed, evacuated, and refrigerated. Preparative centrifugation is classified based upon medium of suspension which the separation is carried out. There are two types depending upon the medium of suspension in which the separation is carried out. In a suspending medium which is homogeneous are known as a differential centrifugation, and in density gradient centrifugation, suspending medium having density gradient are known as a density gradient centrifugation. Differential centrifugation, it is common process in microbiology and cytology. Differential centrifugation is based upon the differences in sedimentation rate of biological particles of a different size and densities. Initially, all particles of homogenates are evenly distributed throughout the centrifuge tube. The tube move down the tube at their respective sedimentation rate during centrifugation. The largest class of a particle forms a pellet on the bottom of a centrifuge tube, leaving smaller sized structure within the supernatant. Fragments are divided into different fractions by stepwise increase in applied centrifugal force. There are some applications of differential centrifugation in bioscience research to separate subcellular organelles, that is mitochondria, nucleus, ribosomes, and isolation of macromolecules such as DNA, RNA, protein, or lipid for further studies. It is used in medicine for diagnostic purpose. For example, purification of influenza virus for studies on fundamental chemical and physical properties of the virus lead to control and prevention of influenza. It can also be used for low resolution separation of the nucleus. As this technique separates particles based on their size, this can be used for the purification of extract containing larger size impurities. There are some limitations. It can damage or contamination and loss of sample takes place. And second one is density gradient centrifugation. It is based on buoyant density. It is also known as equilibrium sedimentation. It employs a medium which has gradients. It is independent on velocity, size or shape of a particle and time. Separation under centrifugal forces depend upon buoyant density of a particle. The gradient, apart from exerting their separating effect, eliminates mixing of a separated component due to convection and mechanical vibration. It is also known at this there are two main types, that is red zonal centrifugation and isopycnic centrifugation. This technique based on basis of their relative velocity. In red zonal centrifugation, the solution have density gradient. This sample has a density that is greater than all the layers in a solution. Density of the solution increases as we move down the column. The sample is applied in this zone at the top of centrifuge tube on a density gradient. Under centrifugal force, the particle will begin sedimenting through the gradient. The particle will begin sedimenting in separate zones according to their size, shape and densities. The different bands are collected by puncturing the tube. There are some applications. It is used for separation of cellular organelles, protein antibody. Antibody classes have similar density but different mass. So separation based on density will not resolve the classes. It separates RNA DNA hybrids, ribosomal subunits, virus particle such as bacteriophage. There are some limitations. The sample occupies a small region of centrifuge tube at the start of fractionation. The sample becomes less concentrated during fractionation. Second type of density gradient centrifugation is isopycnic centrifugation. In isopycnic separation, it is also called as a equilibrium separation. The solution contains greater range of densities. The density gradient contains the whole range of densities of the particles in the sample. Each particle will sediment onto the position in the centrifuge tube at which the gradient density is equal to its own density. In isopycnic centrifugation, separation of a particle occur into zones on the basis of their density differences. It independent on time. Their application: fractionate animal, plant and bacterial cells, viral particle, lysosomes, membranes and macromolecules in a range of process. Commercial separation of viruses for vaccine and immunotherapy products in both batch and continuous flow zonal modes. Next type of centrifugation is analytical and ultracentrifugation. Analytical ultracentrifugation is mainly concerned with the study of purified macromolecules or isolated supramolecular assemblies. The main aim of analytical ultracentrifugation is used to study molecular interaction between macromolecules or to analyze the properties of sedimenting particles, such as their apparent molecular weight. It determined the determination of the purity of macromolecules occurs in analytical ultracentrifugation. The relative centrifugal force of analytical ultracentrifugation is high, that is 6,25,000 gram. The volume of a single tube accommodated 0.2 to 5.5 ml. Their speed is very high. Due to their high speed, the ultracentrifugation needs a refrigerator. The examination of changes in molecular mass of supramolecule complexes. The determination of relative molecular mass of a solute in their native state. The detection of conformational changes, ligand binding studies. The principle of analytical ultracentrifugation. The process: small volume of a sample with a built-in optical system. Molecules are observed optically during centrifugation. Samples are centrifuged in a tube with a quartz windows. As the rotor turns, the images of the protein are projected onto a film of computer. The concentration of protein is determined by absorbance of light. It results in degree of darkness on film or deflection of a recorder of a scanning system. It determines the sedimentation coefficient. Analysis method and application of analytical ultracentrifugation. Sedimentation velocity method. The hydrodynamic properties of a macromolecule are determined by their sedimentation coefficient rate, that the concentration boundary of the particular biomolecules move in a gravitational field. It gives detailed information on complex formation and properties of aggregates. Sedimentation coefficient can be used to characterize changes in size and shape of molecules in varying experimental conditions. It also used to determine purity of molecules. Sedimentation equilibrium method. Sedimentation equilibrium experiment have a lower rotor speed than sedimentation velocity experiment. Solute particles do not pellet at the bottom of the tube, but instead the process of diffusion opposes the process of sedimentation until after a period of time. The two opposing forces reach equilibrium and the apparent concentration profile does not change. At equilibrium, the concentration of a solute increases exponentially towards the bottom. Each column displays a different absorbance profile because the concentration of samples are varied in each. It allows calculation of buoyant density of a particle, molecular size, and aggregation behavior, etc. This is the optical system of an analytical ultracentrifugation. Thank you.